In the year 1700, refined sugar was rarely seen. Once an expensive luxury reserved for the aristocracy, average annual consumption in England was estimated at just 4 pounds per person. The development of massive sugarcane plantations and the European sugar beet industry slashed prices, turning a rare commodity into a global dietary staple. By 1900, intake had reached 100 pounds per person. As the blue curve of sugar consumption climbed, urban centers recorded a simultaneous explosion in diabetes mortality, shown here by the red bars. Industrialization transformed a rare luxury into a widespread environmental pathogen, exposing human biology to unprecedented carbohydrate density for the first time. Before the discovery of exogenous insulin in 1921, a diagnosis of type 2 diabetes was a death sentence. Early physicians had no pharmacological tools to manage metabolic failure. Figures like Sir William Osler kept patients alive through draconian dietary restriction. His 1892 protocol prescribed a diet of 65% fat and just 3% carbohydrate. This raises a difficult question. If the medical community already understood the link between carbohydrates and metabolic destruction, why did global consumption continue to skyrocket? A century ago, humanity possessed the clinical knowledge to prevent the modern metabolic crisis, but that crucial knowledge was systematically overridden. After World War II, a surge in heart disease split the scientific community. John Yedkin identified sucrose as the primary driver of arterial damage, while Ansel Keys blamed saturated fat and dietary cholesterol. This dispute was settled by financial interference rather than data. In 1967, the Sugar Research Foundation paid Harvard scientists $6,500 to author a literature review that shifted the blame entirely to dietary fat. When the 1980 dietary guidelines pushed low-fat foods, the industry faced a palatability problem. To make low-fat products taste good, manufacturers pumped them with massive quantities of added sugars. By institutionalizing a bias against dietary fat, this industry-funded campaign shaped the high-sugar nutritional guidelines dominating global food production for 40 years. The correlation between sugar intake and cancer has led to the popular myth that sugar acts as a direct fuel for tumors. However, systematic reviews confirm that cancer cells are metabolically flexible. They can scavenge lipids and amino acids if glucose is restricted. The danger is the indirect oncogenic cascade. Chronic sugar consumption drives systemic hyperinsulinemia, flooding the body with insulin. This upregulates the IGF-1 axis, hormones that act as biological fertilizers, stimulating nascent malignancies and inhibiting the programmed death of damaged cells. Sugar does not feed tumors directly. Instead, it corrupts the host's hormonal environment, creating a permissive state where cancer can thrive.
High fructose corn syrup is uniquely toxic. Unlike glucose, fructose is processed almost entirely in the liver. It bypasses the liver's rate-limiting brakes. This unregulated influx converts directly into visceral fat, causing rapid hepatic insulin resistance and fatty liver disease. Refined sugar also creates advanced glycation end products, or AGEs. These compounds form when sugar reacts non-enzymatically with proteins, stiffening vascular tissue and triggering severe intracellular oxidative stress. Modern sugars attack the body on two fronts, overwhelming the liver's metabolic capacity while systematically degrading the structural integrity of our proteins. This metabolic damage is particularly severe during the first 1,000 days. From conception to age 2, a child's metabolic programming is highly sensitive to nutritional inputs. A landmark 2024 study utilized a natural experiment in the United Kingdom, the sudden end of sugar rationing in September 1953. During rationing, sugar intake was strictly limited to levels mirroring modern clinical guidelines. Researchers analyzed the UK Biobank to compare adults conceived during rationing with those conceived immediately after sugar intake doubled. The findings were striking. Limiting sugar exposure early in life delayed the onset of type 2 diabetes by 4 years and reduced the total lifelong risk by 38%. This data identifies refined sugar as a developmental toxin that rewires metabolic programming before a child reaches their second birthday. Addressing this crisis requires moving beyond the empty calorie model. Treating sugar as a simple unit of energy overlooks its specific chemical behavior inside human tissue. Refined sugar acts as a chronic metabolic disruptor, driving hyperinsulinemia, liver dysfunction, and cellular decay, regardless of total caloric intake.
the staggering burden of non-communicable diseases requires a shift in focus. Public health policy must move beyond requests for personal moderation and implement aggressive structural interventions to protect the developing biology of the next generation.